talking about molecular evolution today, which is going to be fairly uncontroversial. I'm not going to talk too much about evolution. But when people think about the relatedness of proteins, the relatedness of organisms, similarity of DNA, this whole kind of system, this whole thing is called molecular evolution, how things have changed over time. So I may have shown you this before, but I think this is a funny picture here. This is the Sistine Chapel, right? And here's God touching the creation of Adam here. This is an illustration that some cartoonist has done of the genome touching the mRNA of the cell. Just their interpretation of, I don't know. I actually kind of like this photo because it's silly, but it kind of reminds you that both of these are a creation story, right? And both of these have a philosophy behind them of like, this is where we came from and this is how we originated. One is divine, the other is naturalistic, right? But so what I'm going to talk about today, though, actually is fairly uncontroversial. I'm going to talk mostly about how to align sequences. If you've got two pieces of DNA and they show similarity, how do you actually quantitate that? Right? How do you align those sequences and how do you score them for how similar they are? And if you have multiple sequences, so we just got done talking about population genetics. If you've sequenced 100 individuals in a population, they're all going to have slight variations to them, right? And how do we quantitate that? How do we actually show that graphically so that we can make sense of 100 individuals all differing at multiple places, right? So we're going to talk about sequence alignment, how you align sequences, how you score differences, how you get an overall estimate of what the overall similarity is, and then building trees based on that, graphical representations of how similar things are, right? An alignment is fairly hard to make sense of, right? If you look at this alignment, I've got a bunch of T's, H's, G's, and C's, and this has highlighted where differences exist, but I mean, how do you make sense of that, right? It's just, I don't know, it's kind of overwhelming graphically. So often what we do is take raw data from sequence alignments, and we position sequences in a tree, and the branches of the tree show you how similar those sequences are. So it kind of gives you a graphical representation and a better idea of how to make sense of that data. So I'm going to walk through sequence similarities and tree building, and at the end we'll talk maybe a little bit about how this can be extended into thinking about ancestry and the relatedness of different organisms. Okay. So we're talking about evolution, genetics, and bioinformatics. I'm sorry, bioinformatics. Did I spell that right? Yeah, information. Yeah. Bioinformatics is basically using computers to crunch all of these numbers, right? When we were first starting to do sequencing, our computers weren't very capable of crunching a lot of numbers, right? If you have a smartphone in your pocket, you probably have the equivalent of like a 1960s supercomputer in your pocket, right? We've come a long way in computing technology. So bioinformatics is mostly is comparing different DNA sequences to this vast amount of information that we have stored in databases. We've got hundreds of genomes of organisms sequenced, and all of that sequence data is in databases that are searchable. And you've been doing this in lab this week. So this is bioinformatics, using complex search tools to find similar sequences in this vast genome databases that we have. Okay? So we're going to compare different DNA and different protein sequences. And some of the kind of things we can answer is, how have sequences changed over time? How have sequences evolved? Right? If I've got a population of people, they might actually all be related, right? And I can see in the multiple generations that different alleles may have shown up, and the allelic frequencies may have changed, right? So we can analyze that if we actually have the sequences, and we can talk about how they've changed over time. And then eventually, this may have implications for how related different organisms are. And if we're trying to talk about, I don't know, Drosophila flies, fruit flies, right? Is Drosophila virilis and Drosophila melanogaster, are those actually, do they have a common ancestor in the past, right? If what tells you that an organism is related is how similar its genome sequence is, then how well you can tell the differences between genome sequences is going to tell you how related or how distantly related is that organism from another one, right? So one of the things that bioinformatics can show you, though, is also really important functional things about proteins. So this is a little image that comes from this, this database and this search tool called consurf. This is a conserved domain analysis program. And what it will do is you can take a bunch of different proteins that all are known to do the same function in a cell. Okay, like, I don't know, the first thing that comes to mind is polymerase. Right? You've got a DNA polymerase, flies have a DNA polymerase, and we can identify it as a DNA polymerase by its nucleotide sequence, its amino acid sequence. But when we actually sequence it, the fly and your polymerase may differ in various different amino acids. Well, you can throw an alignment together, and you can align all of these different polymerase sequences. And the amino acids that are identical in every single polymerase is probably a really essential amino acid for the polymerase's function. Amino acids that are different, are not so important, because a fly can still replicate its genome with its DNA polymerase. You can still replicate your genome with your DNA polymerase, but the differences between them are then less significant for the protein's function. Okay? So what this image is, it's a three-dimensional structure, and each one of these little lobes is the amino acid in this protein sequence, and we're showing you the three-dimensional structure of this. Right? And what the colors are is it's coloring the amino acids based on how many copies are present when you put all of these together. Right? How conserved is that amino acid over all of these proteins that we've aligned together. And so what you see is, here's the active site of this enzyme. I don't know what this is, but this is its substrate. This is the thing that the enzyme binds. And you can see around it, all of those amino acids are colored really dark red, indicating that no matter what protein we take, from what organism we take, it always has that amino acid there. It's highly conserved, right? And that makes sense, right? If this is going to do the same function in different cells and in different organisms, those amino acids in the active site are probably not going to be able to vary. Right? The chemistry is very specific to binding this molecule and doing whatever function it is. Right? So that makes intuitive sense. And then ones that are further from the active site are less conserved. Right? In white, those are most of the time conserved. On average, they're there, but sometimes they differ. And then the ones way out, far away from the active site, these guys, it doesn't seem to really matter that much. Those are quite variable. Depending on which organism, they may or may not share a similar amino acid sequence. What's really, now, that seems intuitive and like, well, I didn't have to do a sequence alignment to kind of tell me that, right? Active site things are really important. But what this will tell you is, whoa, that seems really far away from the active site. But every single individual, when I sequence their protein, they always have the same amino acids out there. Maybe that's a really functional part of the protein that you would have missed. And so then you could say, oh, let's see what that's doing. Right? It can give you some really functional information by comparing sequences. Yeah?
That's what my question was.
Yeah. So what do these sequence alignments look like? There's a number of programs that will do this for you. Lots of really smart computer programmers have programmed these tools that will take sequences that you give it and will align them. And basically what the alignment tools are is trying to find areas of sequence similarity. And you've actually done this a little bit in your homework. Remember that homework problem that had you put the sequence on one axis and the other sequence on the other and find where the conserved domains were, or where the identical sequences were? Basically, the computer programs are doing just that. They're taking one sequence and another sequence and putting them together and seeing which, marking which nucleotides or which amino acids are similar in both sequences. But what they do it is over and over and over again, jogging them slightly, introducing gaps, introducing insertions or deletions, and trying to see where the best alignment is over multiple rounds. So you basically did one step in like 1,000 steps that a computer aligning sequence would do, right? So what you end up getting then is something like this. Here's an alignment. It's got half a dozen or more, 10 or more here, sequences. And this is amino acid sequence. All of these have a methionine, so that was probably the start codon, first amino acid. And then it's only showing you a certain number of the proteins, and I've cut it off. There was more alignments that were going on there, but I've cropped the image. And so here, every one of these proteins starts with a methionine, and the second position, most of the time, it's a serine. So those are colored green, showing you the alignments, but sometimes it's an alanine. So what this is showing you is a star at the top means that's a completely conserved, identical sequence all across. Two little dots say that's a fairly important domain, but it varies a little bit. So 2 dots. You can get ones with a single dot. That's not on here.
Yeah, the fifth one.
Fifth. Oh, there you go. Yeah, yeah. All right. So most of the time it's the same, but sometimes it actually has a very different property to the amino acid. So a serine and an alanine have the same basic amino acid chemical properties. So even though they differ at the amino acid level, functionally, chemically, they act fairly similar. So it gets 2 dots, right? Here, 1 dot. Well, sometimes it's an alanine, sometimes it's a serine, but then also sometimes it's a cysteine, which actually has very different chemical properties to it. So most of the time it's the same. It's a fairly important place, but it can vary in chemical properties. Here, I don't get any score because it's got all kinds of different amino acids that can be there. It doesn't seem to be very conserved. Right? So these are what the actual raw data looks like, the alignment looks like. It's a lot of data to look at and to make sense of, though. Right? Now, you can do the similar thing here for nucleotide sequence. Nucleotide sequence is a little bit easier because I only have 4 base pairs to work with, and they all have basically the same chemical property. The only real thing is, well, I got 4 base pairs. 2 of them are purines, 2 of them are pyrimidines. So I could have a purine at the same site or a pyrimidine at the same site. So that might give me a different score. But you're basically one of the 4 or you're not, right? Whereas with amino acids, I can be any one of the 20 amino acids at this site. Okay. So here is a DNA alignment. Here I've got a conserved. Every time I look at this gene, there's always a TCA there. Always an A here. Here, these little dashes, that's where the sequence has actually had to, or the computer program has had to introduce a gap, right? So this sequence down at the bottom, well, actually, let's look at this top one. I didn't have any sequence here, right? There was no matching similarity there. This is where my sequence started. So what it said is, well, I've had to introduce gaps there, and I'm going to start it where I have matching. So a lot of G's are at this site, right? So the computer has made a decision that, well, it's best to introduce a gap there and start way down here at the end. Now down here, this sequence here is 3 base pairs shorter. So what the alignment has done is said, well, to get the best alignment, though, I'm just showing you that there's a gap there. But the actual sequence reads AAT and then right to this next C. But the alignment has just forced a gap. What these tools will do is they'll line it up the best they can, and then they will assign penalty values for how many adjustments they had to make. Right? So if I had to introduce a gap, then I award that sequence a penalty. Right? I had to introduce a gap. I had to introduce 2 gaps, so you get 2 penalties. Now, you don't get as much of a penalty if you got 2 gaps right in a row. Right? I already had to put a gap there. That's the big penalty. If I had to actually make it 3 nucleotides, though, that's less of a criticism against it, right? I already had to introduce a gap, so how big the gap is less important than the fact that I had to put a gap there in the first place, right? So if I had a gap of another sequence and it was, 4 nucleotides, or let's say 6, right? So this one's got 3, the other one had to have 6 gaps in there. I wouldn't say that the one is twice as bad because it's got 6 instead of 3. No, they're both fairly bad because I had to put a gap there. And one is a little bit worse, but not twice as worse, right? So different programs have different values that they assign to this, but you're going to penalize it every time you have to introduce a gap or anytime the sequence doesn't line up properly, right? So like here, I have A's. Well, most of them have T. So whatever most of them have, I don't award a penalty. But if you're in the minority, now I didn't have to put a gap there, but I have an A instead of a T. Well, a penalty has to be given to the sequences that differ than the most common sequence, right? And then you can give penalties if it's an A and a T. Well, that's a transversion, right? Purines are all girls are pure, right? A and G are purines. So if I've substituted a purine for a purine, I don't penalize it as much. But if I had to substitute a purine for a pyrimidine, an A for a T, well, then I award a bigger penalty, right? So basically what you're doing is aligning and then getting scores. So, yeah.
What's a transversion? Well, actually,
I'm sorry. So a transition is when you've substituted a purine for another purine,
okay.
an A for a G, right? Those are both purines, so I say that that is a transition. A transversion is when I've substituted a purine for a pyrimidine, right? So in terms of chemical properties, that's the big difference between nucleotides, is whether you're a purine or a pyrimidine. And then if you're actually a purine, then you could be one of two. But we say that that was a less of a difference. So.
And then I was going to ask earlier, so if they assign like a gap because they can't tell what it is?
No, I mean, we know what the sequence is. Here we know the sequence is AATC. This one, so what it's really doing is saying, okay, everybody is identical there, so I'm going to line everybody up there, because that seems to be really important, right? But this guy is 3 nucleotides shorter, so what it does is it's going to have to introduce a gap in order to keep these things lined up. Now, if it was just 3 base pairs shorter up here, that's no big deal, right? And that we've already done that, right? This guy is shorter, and so we just started aligning him down here, right? But somehow, this guy's 3 base pairs shorter. I'm going to have to decide where to put that gap, right? So the computer is doing multiple iterations of this, aligning it this way, assigning a value, aligning it, introducing a gap, assigning it a value, introducing 2 gaps, assigning it a value. And it's basically picking the best alignment where it has to penalize the sequences the least, right? So what it's done is said, for the least amount of penalty, I'm going to introduce gaps here so that I can keep these ones aligned, because I know that's really important. And I can keep that A aligned, because that seems to be really important, right? I can align these all, and they can all get an A there. So it's trying to minimize the gaps and minimize the penalties that it gives, trying to find the best alignment, finding the most similar sequences all across the entire strand, right? If you think about the computing power this takes, this is a lot, right? The computer has to align it so that T's are all there. Well, but maybe it could shift this over, right? Here's a T that could be aligned over there, right? So if you give it 2 sequences, it's going to try and align them, and it's going to introduce gaps, and it's going to be shifting them all over the place, trying to align them, right? And then if you put a 3rd one in, it's got 3 of them, and it's shifting all of them. And if you put 4 of them in, it's got all 4 of them, and they can all be shifting, and they can all be introducing gaps, right? This is having to do multiple iterations of this. It would take us years to manually do that, like on graph paper, to find the best one, right? But thankfully, we've got really fast microprocessors now, and it can just do this over and over again. Right? All right, so you've got the idea. I'm doing aligning sequences. I'm assigning the sequences a penalty value based on how well I can align them. To make more sense of the data, we usually try and align things. Well, actually, let's talk about, I got one more sequence alignment thing, and then we'll talk about making trees. Whether you're doing an amino acid sequence or a DNA sequence, the rules kind of change a little bit. Because here's here's a codon that codes UUU, codes for phenylalanine, right? In a protein alignment, I would translate that, right? If I'm interested in the protein sequence, I would take the DNA sequence like you did in lab, right? We took a DNA sequence and we translated it into protein, and then we were aligning proteins, okay? So if I align proteins, phenylalanine and phenylalanine line up, but their codons might not actually have lined up, right? If I actually looked at the RNA sequence or the DNA sequence to code for phenylalanine, well, UUU and UUC, if I did a DNA alignment, I would have to penalize these because they're not using the same nucleotides. But if I was doing a amino acid, a protein alignment, then I wouldn't assign it penalty. Phenylalanine meets with phenylalanine. And if I'm doing a protein alignment, I don't care what the nucleotide sequence was. I'm interested in the amino acid sequence, right? We call this a synonymous. So even though there is a discrepancy between the codons, a protein alignment says that's synonymous, right? I translate it into the same thing, and so it would align it and give it no penalties. Right? A non-synonymous change, UUU and UUA, right? I'm differing only by one nucleotide. So at the nucleotide level, I would give these both the same penalty, right? UUU and UUC. Well, I differ by one, so I'd introduce a penalty, right? Here, UUU and UUA, same penalty level, right? Because it's a different nucleotide. But at the amino acid level, I would not give a penalty here because it codes for the same thing. Here, this is called a non-synonymous change. The single nucleotide change has actually caused me to have a completely different amino acid, right? It's kind of like a missense and a nonsense mutation, right? When we're talking about alignments, we just say it's a synonymous or a non-synonymous alignment, right? If it's a synonymous alignment, I would put those two together and say, even though the base pair is changing, I still say that's a good alignment because it's going to give me the right amino acid, right? If it's a non-synonymous change, well, then maybe I shouldn't align those two nucleic acid sequences there, right? Because they give me different amino acids. All right, so that's protein sequence alignment. Nucleotide sequence alignment. Here's a little sequence, GTACCT. And I have a similar sequence, GATCT. Now, to get these to line up, this is probably the best way to do it, because these are not identical sequences. But the best way to do it would be, well, let's introduce a gap there so that I can get my G's aligned. So I introduce a gap. That allows me to get my A's aligned. Now, these guys don't match up, though, right? But it's best to keep those misaligned because that allows me to keep my A's and my C's and my T's aligned, right? So I would have two penalties on this alignment, right? I have a penalty for having to introduce a gap, and I have another penalty, though I would penalize it less because I didn't have to introduce a gap, but I actually have a nucleotide change there. Now, if you think about, if I know that those are like two individuals and they're genetically related to each other, right? Say I took two original male and female flies, I bred them, I got 1,000 offspring, those had 1,000 offspring, and I sequenced two individuals in that second generation, and I found this change, right? I know, because I did the experiment, I know that they're related to each other, right? And so what is the actual change, though, that caused that C and that T to be different from each other? You actually have a couple of scenarios that may have happened in those several generations to cause that change, right? Maybe just the C changed into a T, right? But maybe that C changed into an A in that first generation, and in the second generation, maybe that A changed into a T, right? Whenever you build a tree, you make assumptions about what has happened in the past, right? I know that they're genetically related. I did the breeding experiment, right? But all you're doing is you're just sequencing individuals in that third generation, and you're comparing their sequences. And you are having to infer what actually happened in the past, right? So a tree is a sequence alignment. It is not an accurate representation necessarily of what has happened in the past. You don't know what happened in the past unless you've been sequencing everybody in the past, right? So what this is called is the K value. The K value is the number of substitutions that have happened per site, right? And this is something that you just have to estimate, right? So if you want to be the most generous, the most parsimonious, that is the most simple explanation, you would say, well, in the past, one individual had a C and it changed into a T, right? And so you would assign the K value as 1. One substitution happened. Right? But maybe, here's my scenario number 1, maybe actually in the first generation I had a C, in the second generation it was an A, and the third generation was a T. That gives me a K value of 2. There's 2 substitution events to give me what is happening right now. Okay? The problem that I want you to see is, look at number 5. Number 5 has C and C, right? So currently, right now, those 2 individuals have the same nucleotide. But in the past, maybe they didn't, right? Maybe they originally did in the parents, but in the first generation I had a mutation to a T, but then I mutated back to a C. That is going to, in this case, the K value for an identical nucleotide is the same as the K value for one that has changed, right? What is actually the case? You don't know unless you actually sequenced every individual, okay? So what I want you to just recognize is that whenever you do alignments and you make inferences about what the past genotypes have been, you are doing that. You're making inferences, right? Maybe both of these are true, and really maybe I shouldn't penalize number 4 for being different, because actually maybe number 5 has changed just as much in the past, but it just happens to have changed back so that I can't tell, right? So I just want you to realize that whenever you're building trees, you have that underlying assumption. And what penalties you're giving it may or may not represent actually what has happened to that sequence changing over time. Does that make sense? Michelle?
Wait, so then the K value for number 5 is 1?
It depends on how you assign the K value, right? And that depends on what kind of tree you build. What assumptions you tell your tree building program or your aligning program, what do you tell it to penalize, right? If it's a C to a T, you could say anytime you see just one nucleotide change, give it a K value of 1, right? Unless I have more evidence that maybe it had a number of changes, right? So what I'm saying is the K value is something you assume, right? When you are designing your tree building program or your alignment tool, you're introducing penalties, and I'm saying I should penalize that because it's a change, right? If you're thinking about how this sequence has changed over time, then I'm assigning that as saying there was a change there, but there was only one change. But maybe actually it's that there was 2 changes or maybe 3 changes. Or places that don't seem to have changed, maybe they have been changing, right? You're limited by what you assume has happened in the past. Now, usually the best thing to do is just pick the simplest explanation, right? But that might not actually, the simplest explanation is not always the one that's true, right? So you're just assuming that. So when you build trees, you're trying to graphically represent how similar are these 2 sequences to each other, okay? And I'm intentionally making my trees really simple here. If I just have 3 sequences and I line them up and I give them a score and I say, this one matches up with this one in a certain way and I give it a score, right? This one matching to this one, I have to penalize that alignment so many times because of gaps or substitutions or changes, right? If I took a 3rd sequence, then I would line it up with number 1, I would line it up with number 2, and I would assign it a score for each of them. And a tree then takes 3 sequences and it aligns them in this graphical representation, and the distance of the arms indicates how similar they are to each other. So in this unrooted tree, what I'm saying is basically sequence number 1 differs from sequence number 2, but if I would trace the distance along here, the distance along is basically a representation of how good the alignment is, or conversely, how bad the penalty is, right? So it's got a certain penalty value associated with aligning those. But if I look at sequence 1 and 3, they are basically the same distance apart from each other, right? They're grouped together in the tree because we have found some significant alignment, but the penalty between 1 and 2 is the same as the penalty if I lined up 1 and 3. And the penalty for lining up 2 and 3 is the same there. Right? So this tree is just a graphical representation saying 1, 2, and 3 differ. They differ in unique ways, but the penalty that I have assigned them for their differences is identical. So they are similar sequences. They differ from each other. They differ from each other in different ways, but the penalty I have assigned to the differences is the same. So that's an unrooted tree. I don't know which one is to give more weight to, which one is the one that actually more represents the sequence that, like it was in the past. I don't know. I'm just saying those three guys are similar. They're similar in this defined quantity way, right? Now, if you're going to root a tree and actually give somebody priority, you usually have to take an out group, somebody that you know is genetically related to this group, but you know is related in a more distant manner. Right? I'm going to pick an uncontroversial example and say dogs. Right? So say I'm doing dog analysis of a protein that's found in dogs. I might survey, my dogs that I've been breeding, right? I've got a female, I've got a male that I want to introduce, and I sequence them and look at their difference in protein sequences. If I just take 3 dogs and compare them, I don't know who to give priority to, right? But if what I do is then go back to the wolf, which we have really good evidence that the gray wolf is the originator of the domesticated dog, right? So somewhere in human history, somebody started keeping wolves and breeding them selectively, and that's what we have now is the common dog. That's the same species. Canis lupus is the wolf. Canis lupus familiaris is the subgroup of what we call the domesticated dog, right? So you take 3 domesticated dogs and you compare their sequences. I don't know who's more closely related or what sequence is more representative of what the original wolf sequence was. But if I actually do introduce the wolf sequence and I say, oh, the wolf sequence is number 3, and 1 and 2, these are domesticated dogs, I can then root my tree, right? Whoever is more closely related to the wolf, I can say, well, they are the ones that had a relation in the past, right? So you root a tree by an outgroup, somebody that you know is related, but is known to be more distantly related than the ones that you're trying to compare, right? Now, sometimes people make assumptions about relatedness, and so we'll make trees of all apes, and we'll take an outgroup of a different kind of monkey to compare to the apes, right? So there are often philosophical inferences that are made about who is a reliable outgroup and how should I use to root this tree. So there's philosophical assumptions that go on when you decide, I'm going to give weight to this outgroup in order to root my tree at some distant place in the past, right? An unrooted tree doesn't tell you anybody, it doesn't give anybody preference for being the historical person. A rooted tree says, the wolf is the more historic, and then anybody else is an outgroup from that. Do you have a question?
Yeah. What's the A represent?
The A represents a theoretical individual in the past that was a common ancestor to these.
Okay. So when you're saying
Of this historic dog-like thing in the past.
So
right?
you're trying to take a dog that you have right now, and you're trying to compare it to another dog, and then you're trying, in order to make a tree rooted, you have to compare it to what you knew as a historic ancestor?
I'm comparing a current wolf with a current dog with another current dog. But I say, I know for lots of other reasons that wolves and dogs had a ancestor in the past. There was some historic wolf way in the past that some ancient man domesticated, right? And so I know that the wolf is the wild version of that, and that these two dogs are more closely related to each other than this wolf and any of those two dogs are.
Okay.
The ancestor that they had in the past is further away in the past. Right? That allows me to root it. I'm putting some historical information into it by saying that ancestor in the past, these two domesticated dogs I know came from this same lineage, right? Those two domesticated dogs had in their lineage some guy that was originally from that domesticated dog. This one is more distantly related than these other two, right? I would not put number 3 in here with number 2 because historically I know 1 and 2 are more related to this historical dog that was domesticated. And that domesticated dog was related to this original dog that was the original 2 dogs that were domesticated, right? So it's giving you historical information there.
And is the historical information from their genome, from their comparisons?
We have several lines of evidence that say that the domesticated dog came from the wolf. I mean, we have like historical, descriptions of people's dogs back in the past, and then we do have genome sequence. So that information about rooting it could be various. It could be other trees that somebody else has built with all the assumptions that came along with those trees, or it could, if we're talking about you and I breeding finches together, like I could tell you, look, I've been breeding this finch, and I've been breeding this finch. So it could be actual observed data that allows you to root it. That's going to be the most reliable. Or it might be other genomic data. And so then that all has its own assumptions too, right? So depending on who you're talking to and what their assumptions and what data they're basing it on tells you how reliable or how much weight you should give to their outgroup, right? I just want you to see there's a whole load of assumptions about assuming who the outgroup is. So I picked dogs because we're pretty sure, right? But if you're going to root dog with foxes, right? We have not, nobody has recorded anything in history about whether or not dogs or foxes were related and people saw them differentiate, right? So the evidence is other molecular data.
Okay.
So it's all like building on itself.
In the book, they have these trees, but then they place the specific people based upon their percentage of relatedness on their frequency, or I forget what it said specifically.
Yeah.
They're basically saying, look, 1 and 2 are 98% similar, but 3 is 45%
Right.
similar.
So that would be the outgroup.
Right. So
And that then roots your tree, right?
Right.
So,
Okay.
yeah. Now, if you really believe that that's a true root, you'd have to have some other historical evidence for that too, right?
Okay.
Because then it's just a if you say, I'm going to build my tree based on percentage likelihoods, well, that's just kind of an internal rule that you've applied to it, right? Somebody else might say, I don't buy that assumption. I don't think those changes are as significant as other changes, and so they might build a different tree using that same evidence, right? So when you determine to root a tree, you're doing it for some reason, or maybe it's a good one, or maybe it's a bad, and people disagree about this all the time. All right, let me talk about, in the last couple of minutes, the types of trees that you can do and what assumptions they make. So I just want you to know the definition of these. A neighbor joining tree. So this is exactly what we're talking about. What are the rules of how you set this up? Determine what your tree is going to look like. A neighbor joining method says, okay, I've got sequences A, B, and C, and I'm comparing them with B, C, and D. Right? So I'm comparing sequence A to sequence B, sequence A to sequence C, A to D, and then same thing. B to B, that's going to be identical, so you throw that out, right? That's obvious. What this does is says, okay, I could align A and B, and I gave it a very low penalty score, right? I was able to align those very well, introducing very few penalties. So the neighbor joining tree says, okay, we should start with those then. Those must be the most closely aligned sequences. So you make your tree and you say those are the case. Then every subsequent calculation after that, the computer program says, okay, it does a screening of them all, gives them all assignments, and it says, okay, A and B are the best, And so then what it does is it makes a composite sequence of A and B sequence. And it says then, okay, how close is C to both A and B? And it assigns it a score off of that. And then it says, okay, how close is D to both A and B? Right? It basically takes A and B and treats them now as a unit. Right? I've said they're the most similar, and so the composite sequence of both of those, how close is C to it? And then you make your tree off of that, right? So when you do that analysis, you would say, okay, well, C is closest to the AB hybrid. So I put that one next. And then I would make a hybrid of A, B, and C. I would have a compiled sequence that says my sequence could be any of the various alleles that represent any of A, B, or C's sequence. Compare that to other things, right? So it starts with the closest joining neighbor and then kind of compiles those into one sequence, compacts those into a one conserved sequence, and then compares everybody else to that compared sequence, or the compiled sequence. Does that make sense? When I assign the alignment a score, I say, how close is C to A sequence or B sequence? I don't penalize either one, right? As close as it is to either one of them is what I'm aligning. So then it would say, okay, C was better at becoming at a similar sequence to both A and B. It was better than D, so I root that. Now I take an ABC amalgam and I compare it to everything else. So it does it multiple generations analysis, okay? Which biases your tree, right? Because it says, well, that was the historical thing, that's the most important thing, was that first alignment. Maybe that's not true, and maybe we should have rooted somebody else, but that's what a neighbor joining says is the most important thing to do, okay? A parsimony tree. A parsimony tree says the number of changes that have to have happened, that's the most important penalty value to assign. How many changes must have happened in this whole tree that I build? I'm going to take multiple sequences, I'm going to build the whole tree, and I'm going to limit how many nucleotide substitutions had to have happened to get that tree. Okay? So here is position number 1. Position number 1 in my sequence alignment here is a G. Everybody's got a G. That's an easy tree to build, right? So on the branch ends, that's the actual sequence I'm dealing with, right? I have to align 4 sequences, so I have to have 4 branch ends. What it says is, okay, well, sequence 1 and sequence 2, those had a common ancestor, it had a G. Those had a common ancestor, it had a G. And the common ancestor of that had a G, right? No matter how you build your tree, that's the case, right? Everybody's got a G, right? Now look at site number 2. I've got one person differing. No matter how you build your tree, what you're going to do is say, well, T is the most common one, so T must have been the ancestor, and at one point there was a branching off, right? One substitution occurred. Now every tree looks slightly different, right? If this is sequence 1, 2, 3, or I'm sorry, sequence 1 is a C, right? So that's C. But is it most closely related to the T in number 2? Or is it most closely related to the T in number 3? Or is it most closely related to the T in number 4? You could build your tree any one of those ways, and the parsimony tree would say, well, assign them all the same value. Right? Where a parsimony tree comes in is where it picks the simplest explanation. And that's easily seen in site number 5. So in site number 5, I've got 2 instances of T and 2 instances of C. I could build my tree that says, well, the ancestor was a T, and one person has the ancestral, the other sequence has the ancestral, but 2 people in the past have had mutations where the T has changed to a C. Right? That tree explains the phenomenon, right? Is it true or not? I don't know. But a parsimony tree says, no, that's not the tree we should build, because that introduced two places where a mutation had to have happened. A parsimony tree will draw you a tree that says the common ancestor had neither of those, right? Two of those come from a common ancestor with a T. These two come with a common ancestor from a C, and the previous ancestor had something else, and so there was one change, right? So the common ancestor either had a T, and when that common ancestor split, there was only one change that was made to give you a C, right? So this says that the change happened further back in time, and it only happened once. And what you're seeing as duplications is just families growing up, having the same genotype, right? So this says the mutation was earlier in the past, and two individual mutations account for it. This one says further back in the past, a single mutation happened, and that gives rise to the variation that we see. The parsimony tree says that's the simplest explanation, so that's the one I'm going to go with. But notice this doesn't have any no basis in historical reality, right? Maybe it happened either way. We don't know. So whenever you build trees, the rules that you've told your program to build the tree off of is what it's going to tell you. So it's always just an estimation of what happened. Right? Maybe the case is it was an A, it changed to a T, then it changed back to a T, and then it changed back to an A, and then to a C, right? We don't know what happened in the past. All you're doing is making the most simplest explanation for what could have happened in the past. Does that make sense? All right. I'm sorry, I'm going way over time. I won't even tell you the maximum likelihood. Well, okay, I'll tell you. Yeah. Maximum likelihood says, yes, things occur in the past, and we have some idea of how frequently they occur. So the one thing I just want you to know, it considers the probability of those mutations actually happening based on what we know about how mutations work. In transitions, the change from a purine to a purine happens more often, just as we have empirically seen as we've looked at mutations occurring. Mutations that go from like an A to a G, a transition, both a purine, those seem to happen more frequently than transversions do. So you can build that probability into your tree making program, right? So you will penalize sequences that have transversions more than you'll penalize sequences that have transitions. So the maximum parsimony said, let's just pick the easiest one to happen in the past, right? Where only one thing had to happen. But the maximum likelihoods tree says, well, that might not tell you exactly. We know that A's and G's more often mutate. So we'll build our tree so that we minimize the number of transversions and maximize the number of mutations that were just transitions. Depending on which tree program you build, what gene you look at, and how many individuals you look at, you can get widely different trees based on how much stuff you put in there, right? So can you actually create a universal tree of life and trace back everybody's ancestors? You can try, and people do, and they make trees, but they differ depending on what kind of assumptions you make in the tree building program, what gene you use. Right? If I'm going to align all of the eukaryotes, all of the archaea, and all of the bacteria, and try and align those all together, I have to pick a gene that we all have, right? Which gene do we all have? Well, we have mitochondrial DNA, we all have cytochrome C, we all have ribosomal RNA. But based on which gene you pick, you'll get very different trees coming out of it. So even diehard committed naturalists disagree vehemently on which genes you should use and what the actual tree of life should look like. No matter who you are, you have to recognize no one was there, we don't know, and we're just making our best guess based on our best assumptions, right? So you are not your genes. No one has inherited a messy gene. Your behavior is your own. And as Christians, we all believe that we have souls, right? You are not determined by what genes you have, right? You actually have some immaterial aspect to yourself that allows you, and this is mystical, but we're mystics, right? Allows you to, in a sense, transcend your genes, right? You can decide whether to clean your room or not, despite whatever genetic proclivity you have, right? Maybe there is a genetic tendency to not clean up after yourself. I don't know, neurobiology, genes of some sort. It doesn't mean you shouldn't, tidy up, right? So you are not your genes.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.